aspects of genetic architecture, including the genetic architecture of multiple traits and pleiotropic associations across the genome. I have nothing to disclose. So the subject of today's talk is pleiotropy, which is the phenomenon that one gene or genetic variant will affect multiple diseases or traits. And the reason that we are talking about it is that pleiotropy is central to the biological interpretation of genetic associations. Quite often, when you identify a disease-associated gene or genetic variant, one of the first questions you will ask is what other traits it is also associated with, because if it is associated with some other related traits, or even sometimes if it isn't, that naturally suggests mechanistic hypotheses. You can also learn quite a lot by looking at the pattern of pleiotropic associations across many loci, genome-wide, about the relationship between different phenotypes. I'll be focused mostly on common diseases and complex traits today, but it's worth noting that pleiotropy, the term, means a slightly different thing in the rare disease world. So when you're talking about rare diseases, oftentimes it refers to these sort of syndromic effects, where a patient will come in with a genetic mutation in a gene that's maybe expressed in different tissues and different organs, and as a result will present with multiple different phenotypes. In the common disease context, it's not as essential that you observe multiple phenotypes in the same individual. So instead, you might have a genetic variant that affects multiple autoimmune disorders, but you won't see one individual with multiple diseases. Instead, you'll see that it has different consequences in different individuals depending on other genetic factors and other just maybe environmental or random factors as well. So today's focus is going to be mostly on common diseases. There's also this distinction that I alluded to earlier between pleiotropy at a locus, sort of as a means of identifying the mechanism of a locus, as opposed to pleiotropy as a genome-wide pattern. So at a locus, you can understand a lot about what a gene or genetic variant does by looking across traits. And if you're looking across a large number of traits, this style of analysis is often called PheWAS, phenome-wide association study. That's not going to be the subject of my talk. Instead, I'm going to be focused more on genome-wide pleiotropy, which is addressing the question of what is the relationship between different traits and looking at the pattern of associations that are shared. The talk will have two parts. I'll begin by discussing genetic correlation, which is really the starting point for any investigation of pleiotropy, and local genetic correlation, which is especially important if you're interested in molecular phenotypes. Then I'll talk about causality and Mendelian randomization, which is, I think, a topic that a lot of people care about because it really gets to the heart of why we do human genetics. For those of you who are just coming in, do feel free to come sit in the front. So if you want to quantify the amount of pleiotropy between two traits or between multiple traits, really the starting point is to estimate a genetic correlation. And this is the only slide, by the way, that has equations on it. You'll probably recognize the first equation, which is just the standard linear model of a quantitative or a binary trait. And it just says that we can partition the phenotype Y into a heritable component, X times beta, plus an environmental or just a non-heritable component, epsilon. And then instead of computing the correlation between two phenotypes, like Y1 and Y2, we can just compute the correlation between their heritable components. So X beta 1 and X beta 2, and that's the genetic correlation. Oftentimes, you'll hear genetic correlations described as a correlation between effect sizes across the genome as opposed to a correlation between heritable components across individuals. And that's kind of a subtle distinction. It's not wrong to describe a genetic correlation that way, but I will highlight a couple examples in the slides that follow of how those two sort of notions of a correlation can differ from each other. As to how a genetic correlation should be interpreted biologically, let's consider a famous example of a genetic correlation, which is that between height and coronary artery disease. So there's this well-known phenotypic association where individuals who are shorter are more likely to have a heart attack later in life. And it's an important question, sort of the reason for this association, for this correlation. Quite plausibly, it could be driven entirely by confounding factors, or really I should say societally mediated factors, socioeconomic status, diet. But this study, published in the New England Journal, looked at the genetic correlation between these two traits. It didn't actually compute the genetic correlation, but it basically looked at the genetic correlation and found that there's a shared biological basis for these two traits as well, where genetic variants probably affect some growth and development pathways that affect adult height and then also affect coronary artery disease risk later in life. Now, it's worth noting here that genetic variants will also influence societally mediated factors like socioeconomic status. So it's not the case that genetic correlations are unconfounded in any real sense of the term, but nonetheless, you can get a bit closer to the underlying biology that's shared between two traits by computing a genetic correlation. Across traits, the interpretation of the genetic correlation can differ substantially. Some pairs of traits, and I'll talk about this more in the second half of the talk, will have a causal relationship. So that's the case for triglycerides and coronary artery disease, which do have a causal relationship. More often, you have some shared pathways or shared biology, which is the case probably for height and CAD, as well as for bipolar disorder and schizophrenia, which are highly genetically correlated. Some traits are so closely related that it no longer really makes sense to describe their genetic correlation as being the result of shared pathways. So adult BMI and childhood obesity are an example of a pair of traits that are just so obviously related to each other that you should just call it something different. And then some cases are kind of complicated. There's a pretty substantial positive genetic correlation between autism and educational attainment, a positive correlation. And the reason this is complicated is that both phenotypes are so heterogeneous. So autism is very heterogeneous phenotypically. Some individuals have intellectual disability and of course will have lower educational attainment. Others are gifted. And similarly, educational attainment itself is a heterogeneous phenotype. It's thought to have a cognitive component as well as a personality-related component. And it's not wrong to say that they have shared pathways, but it's just not the whole picture. There are sort of other questions that are relevant to the interpretation of this particular correlation. I should also mention that assortative mating can produce a genetic correlation between two traits. And this is relevant to the distinction that I described earlier between the correlation between genetic components as opposed to that between effect sizes. So plausibly, you could have two traits with no pleiotropy, no shared causal variants. But if individuals with one trait are more likely to mate with individuals with the other trait, then their offspring will have a correlation between the genetic components, right? And so this is just a way in which you can get a genetic correlation without actually having any shared biology. Now, I don't really think myself that this is a compelling explanation for most of the genetic correlations that we observe. I think that most of them do represent genuine shared biology, but it definitely can affect the genetic correlations that we observe. This plot is showing, by the way, the correlation between mates in the phenotypic value as opposed to the genetic correlation on the y-axis. They're highly correlated, but what you might observe if you're able to read the axis labels is that the correlations on the x-axis are much weaker than the correlations on the y-axis, which is part of why I think that most of these correlations do represent some shared biology, even if they can be inflated or influenced by this effect as well. If you want to estimate the genetic correlation between two traits, usually the starting point is LD score regression, and there are other methods that do this as well, including more recent methods that have better statistical power. But LD score regression has the particular advantage that it only requires GWAS summary statistics. So this is a distinction that many of you will be familiar with. Methods that operate on GWAS summary statistics as opposed to individual level data have advantages, and they're very convenient. It's especially convenient when you're looking at the relationship between different traits that you might not observe in the same study. So you might have one study for one trait, another study for the other trait, and you can't even compute a phenotypic correlation because you just didn't observe the phenotype in any of the same individuals. But you can get around that using LD score regression and using the fact that you have genetic data for individuals in both studies. All of the genetic correlations that I've been describing so far have been restricted to common genetic variants. But there's also this question of whether the genetic correlation differs between variants that are common and variants that are quite rare in the population. And we recently published a study where we characterized the genetic architecture of rare protein coding genetic variation. And one of the questions we were interested in that study was whether common and rare variants are mechanistically convergent, whether they seem to implicate similar genes and pathways and cell types. And one of the findings from that study was that we looked at the genetic correlation between various pairs of traits, restricting to common variants or restricting to rare variants. So in the upper triangle here, this is the common variant genetic correlation estimated using LD score regression, and in the bottom triangle is the rare variant, the genetic correlation of rare loss of function variants, protein coding variants, estimated using a new method. And these are highly concordant, suggesting that whatever the pleiotropic processes are that are producing these genetic correlations, those pleiotropic processes are modulated both by common and by rare genetic variants, leading to these concordant genetic correlations across traits. Interestingly, and I don't know what happened here. There's supposed to be points on this plot, but this is you can imagine there's sort of a cloud of points following this dotted line, not following the y equals x line, which is solid. The rare variant genetic correlations are a bit stronger on average than the common variant genetic correlations. We're not sure why that is, but we think it might have to do with pleiotropic negative selection, where pleiotropic variants are less likely to become common in the population because they have larger selection coefficients. Just as you can partition the genetic correlation into a common variant correlation and a rare variant correlation, you can also partition it across the genome. So when you look at just a locus, typically a couple megabases, the genetic correlation restricting to SNPs in that locus is called the local genetic correlation. There's a method called HEST that estimates this, as well as other methods. And the reason it's useful is that it provides insight into genome-wide heterogeneity. So this is a plot for HDL and LDL cholesterol levels, which are two closely related traits, but actually they only have a weak negative genetic correlation. It's pretty modest. And when you look at the local genetic correlation across the genome, what you observe is that actually there are some regions of the genome with positive local genetic correlations and others with negative genetic correlations. That's being shown here. It's not the red and the blue, which is just the chromosome, but rather whether it's going down or up. So most of them are going down, but several of them are going up, indicating that you have quite a bit of pleiotropy that's kind of masked because it approximately cancels out the positive and the negative effects when you look just at the single number, which is the genome-wide correlation. And the local genetic correlation is also an important concept to understand if you're interested in molecular phenotypes, such as gene expression. So a gene is, at a locus, you have some TSS, and you have some window around the TSS where you expect to find EQTLs. And a very commonly used approach when you identify a non-coding GWAS association is to ask whether that non-coding variant is an EQTL for some gene, and in particular to perform TWAS, which stands for transcriptome-wide association study, to identify putative causal genes. TWAS should be understood as measuring the local genetic correlation between a phenotype of interest and the expression level of some gene. And it comes with all of the same caveats as other genetic correlations. I'll talk more about causality in just a couple slides, but it's worth noting that just because you see a genetic correlation or a local genetic correlation, it doesn't mean that there's a causal effect of a particular gene on a particular phenotype. And sort of one particular way that you can get a spurious TWAS result, but this is also applicable to other local genetic correlations, is when you have different causal variants that are in LD with each other. So oftentimes you have, you can have one causal variant for one trait, another causal variant for another trait. Maybe they're close by and they're in LD with each other. If that happens, then you'll often get a genetic correlation between those two traits, even though there's no shared causal variant. So this is another sort of way in which the correlation between the effect sizes, which in this case would be 0, can differ from the correlation between the genetic components. We don't worry about this as much genome-wide because genome-wide there's it's likely that these types of tagging effects are going to cancel out. There's no reason to expect they're going to be systematically positive or systematically negative. But at a locus where there's just a few causal variants, there's no reason to expect that they would cancel, and so you can get some sort of spurious correlation. This is a study, Chun et al. What they did, I really like this paper. They looked at a large number of loci, autoimmune disease loci, that seemed to overlap with EQTLs in white blood cells. And what they found is that at most of those loci, those basically TWAS loci, there wasn't any evidence for co-localization. In particular, there's no evidence that the causal variant is actually shared. In most regions, it looks like it's related to LD. This is an example of a locus where actually it looks like the causal variant is shared. So these are 4 Manhattan plots that all have nearly identical shape. These top 3 are for 3 different autoimmune diseases. It's not surprising to see that there's co-localization between the autoimmune diseases because we know there's lots of pleiotropy among these particular traits. And then also the Manhattan plot looks identical for this, in this case it's EQTL and CD4 plus T cells. Now let's talk about causality and Mendelian randomization. This is sort of the first thing that a lot of people think about when they start thinking about pleiotropy, just because everyone wants to identify causal relationships. It's a big part of why we do human genetics, is because it's kind of causally anchored. And this has been a really important approach in the field. The motivation is that observational studies are very easily confounded. This is probably something that everyone in this room kind of understands. In an observational study, you basically correlate some exposure of interest with a health outcome, and you typically just try to account for every confounder that you can. So you ask people all sorts of things about their, where they live and their socioeconomic status and whether they smoke and all of these things. But there are always going to be unobserved confounders and more generally measurement error in the exposure as well as in the covariates that you're trying to account for can cause these associations to be spurious. There's many other issues as well, but really the main motivation for a genetic approach is the difficulty of accounting for unobserved confounders in observational studies. Mendelian randomization is an approach that's been proposed to solve this problem, and the idea is as follows. You have some biomarker or exposure, in this case it's LDL cholesterol levels, a health outcome, in this case it's coronary artery disease, and a genotype that affects the biomarker or the exposure. So in this case it's a genetic variant in LDLR, and the idea is that if you can exclude the possibility that genetic variants in LDLR affect coronary artery disease through any mechanism besides LDL cholesterol, then any association that you observe between the variant and the outcome must be mediated by its effect on the exposure, and you can use that association to back out an estimate of the causal effect. Now, if this assumption that the genotype is not associated with the outcome through any sort of pleiotropic or other means seems like a strong one, it is. And in fact, it's a big problem in Mendelian randomization studies that genetic variants pervasively have pleiotropic effects that might not be mediated by any particular trait that you observe. Nonetheless, there have been quite a few compelling examples of Mendelian randomization done right. And I'll just show you just a few of my favorites. One is that in the context of coronary artery disease, we know quite a lot about what the likely confounding factors are. So if you're thinking about lipid fractions, really the main players are LDL, HDL, and triglycerides. There could be others too. But really, we think that if you can account for the effect of those 3 things, you've probably done a pretty good job of accounting for potential pleiotropic effects. And this was a study that looked at genetic variants that are associated with each of those traits and that are associated with different combinations of them. And what it found was that LDL and triglycerides are probably the causal factors for CAD, in particular not HDL. That's consistent with evidence from clinical trials, but it's different from evidence from observational studies, which have tended to find that HDL instead of triglycerides is causal. And the reason for that is measurement error, which I mentioned a couple slides ago. Measurement error in triglycerides, which are very unstable, they spike if you put cream in your coffee, causes HDL to actually be a better proxy for like steady state triglyceride levels compared with the triglyceride level itself. So like it's a slightly similar story as like blood glucose and HbA1c in T2D. Another example that I quite like is this study of vitamin D levels in multiple sclerosis. The motivation here was that it's well known that individuals from higher latitudes are more likely to develop MS because presumably, at least the theory is that it's because they get less sunlight, they have less vitamin D, and that causes increased risk of MS. And this study looked at 4 genetic variants in these 4 genes that are known to be involved in vitamin D production and synthesis and transport. And what they found was that these variants are associated with vitamin D levels, and they have consistent associations with MS risk, all 4 of them, suggesting that vitamin D is actually causal. And what the key thing here was that because they actually have some mechanistic information about how these variants affect vitamin D levels, it gives them a lot more confidence that the effects that they're seeing on MS are not mediated by some other factor. And then this is my favorite example, which is ALDH. So ALDH genotype affects alcohol metabolism, and it's quite common in East Asian individuals to have poor alcohol metabolism as a result of this genotype. I'm actually heterozygous for this myself. And it's strongly associated with alcohol consumption because you suffer more ill effects. You feel bad if you drink too much as a result of this genotype. And that provides the opportunity to investigate the effect of alcohol consumption on other phenotypes, in this case, blood pressure. The authors of this study found, this is actually a meta-analysis, but of these studies looked at individuals mostly in Japan, in places where basically men drink and women don't. And what that allowed them to do was to rule out, or at least partially rule out, a pleiotropic effect. So what they observe is that ALDH is associated both with alcohol consumption and with blood pressure in men, as you'd expect, if there were a causal effect of alcohol consumption on blood pressure. But there is no effect either on alcohol consumption or on blood pressure in women, which is encouraging because if there were a pleiotropic effect, unless if it were some sex-specific effect, you would expect to see some sort of vertical relationship here, where these variants are associated with blood pressure in women as well. Despite the fact that I really like these examples, I do think that most applications of Mendelian randomization are problematic. And Mendelian randomization can easily be applied, especially when it's applied kind of out of the box, in a way that just gets the wrong answer because of violations of the sort of no pleiotropy assumption. What we did, this is a study from a few years ago now, is we applied Mendelian randomization kind of out of the box to 429 trait pairs that were ascertained on the basis of their genetic correlation. This is just 429 nominally significantly correlated trait pairs. And what we found was that MR turns up a causal effect for 271 out of the 429 trait pairs, which is not really plausible, including a bidirectional causal effect, so A causes B and B causes A, for 155 of them. Now, this was in 2018, and new methods to account for pleiotropy do help, I think, at least to an extent. There's a method I like called cause. There are bidirectional approaches, which help quite a lot. There's a method called MR-Presso that tries to get rid of outlying genetic variants, so at least you can see that the correlation you see isn't just driven by just a few variants. There's a weighted median approach, which has the same intuition, and also Steger filtering is an important approach that helps especially with these bidirectional associations. We developed a method called the latent causal variable model, which aims to quantify genetic causality continuously. And the motivation for this is as follows. Instead of drawing an arrow between like Y1 and Y2, we're just going to assume that the genetic correlation between Y1 and Y2 is mediated by something that we don't observe. So a special case could be the genetic correlation is mediated by Y1, in which case that means that Y1 is causal for Y2. But more generally, there could just be some unobserved causal factor, in this case it's L, which causes this genetic correlation by affecting both traits. And we define this kind of partial genetic causality which refers to this phenomenon that like maybe L isn't exactly Y1, but it's closely related to Y1. So an example of this might be that you have BMI, turns up as being causal for all sorts of things, but maybe it isn't exactly BMI. Maybe there's some unobserved adiposity phenotype, which is actually the causal factor, and BMI is really closely related to that phenotype because like most variation in BMI is due to variation in adiposity, not because some people are bodybuilders, but maybe it's actually that other phenotype which is really the causal thing. So that would be an example of a partially genetically causal relationship. And this method sort of shares the intuition with all the other sort of robust Mendelian randomization methods, which is that you can distinguish between genetic correlation and causality by looking for some sort of consistent pattern of effects across many loci. So in the absence of a causal relationship, you'll observe some variants that have shared effects on both traits, but also other variants that affect, in this case, trait 1 but not trait 2, and variants that affect trait 2 but not trait 1. When you see that, it means there's no causal relationship. But what a causal relationship looks like is that even though you're going to have variants that affect trait 2 but not trait 1, all of the variants affecting trait 1 sort of have proportional effects on trait 2 as well, and that's how you can tell that there's a causal relationship. Obviously, when we have noise and we look at real data, it never looks like this, and that's why we need sort of sophisticated statistical methods to identify these types of relationships. And when we applied LCV to this set of 429 trait pairs, we identified a much more sort of plausible number of partially genetically causal associations, which is 30 trait pairs. And this is what that looks like. You can see that there's tons of genetic correlations in this plot, which is these pluses and these minuses, but it's much more sparse, the causal effects, which are here in blue. And for example, for CAD, we identify 5 traits that are affecting it causally: BMI, LDL, triglycerides, high cholesterol, which is closely related to LDL, as well as hypothyroidism, and HDL is not estimated to have causal effects. So really the important punchline from this, I think, is that most pleiotropic relationships do not reflect causality. And I think it's a really important sort of future direction for the field to try to understand the source of all of these pleiotropic relationships without relying on the notion that we're going to identify specific traits that are causal for specific other traits. I think most of the time, the things that are causal are just not observed in our genetic association studies, and we just have to have better methods and just maybe better ways of thinking about the genetic association data in order to understand what those unobserved processes are. And I'll just share sort of one piece of intuition, for how we might understand some of these sort of pleiotropic relationships, which is that variants with similar effects across multiple traits. So pleiotropic variants that maybe affect this trait, increase this trait and decrease that trait, may act via similar pleiotropic mechanisms. So if we want to understand what those mechanisms are, maybe a starting point will be to identify subsets of genetic associations that look like they have similar effects across multiple traits. And there are a number of methods that sort of use this same intuition, including one that we developed called pleiotropic decomposition regression. And this is just an example of what that looks like, where we have some 3 heritability components comprising different subsets of SNPs that affect asthma. So you can see all 3 of these components explain about 1/3 of the variants of asthma, the heritability of asthma. And one of them is closely related to BMI. That's consistent with the fact that there's an obesity-related asthma subtype. One of them is most closely related with pulmonary function, as measured by FEV1 FBC, and one of them is most closely related with inflammatory phenotypes, especially eosinophil counts. So just in summary, I've told you about how pleiotropy is central to the biological interpretation of genetic associations because it tells us quite a bit about mechanism. It can be measured using the genetic correlation, although we've seen that different traits have quite different interpretations for their genetic correlation. Common and rare variants have similar genetic correlations, and that's interesting because it suggests that they're mechanistically convergent, at least at the level of pleiotropic pathways. And local genetic correlation is an important concept in order to understand genome-wide heterogeneity and molecular phenotypes. We've also talked about Mendelian randomization, which is powerful in special cases, but problematic when applied out of the box, and the fact that most pleiotropic relationships do not reflect causality. I'll just wrap up by saying that I am hiring, so if you're interested in this work, then please do feel free to come and introduce yourself or send me an email. And thank you very much for your attention.
Thanks, Luke, for this excellent educational introduction to this topic. Are there any questions from the audience or online? Yeah, please go ahead. Use those microphones which are nearest to you. You can already queue up there if you want. Thanks. Please go ahead.
Yeah. Okay. Hi. Thank you. This is Xiao from AstraZeneca. Thank you very much for your very nice presentation. Just one question about from your summary. It says common and rare variants, they potentially have the same genetic correlation. I just wondering if you have any idea of how we address the potential pleiotropy for the rare variants.
So that's a good question. So first of all, the fact that the genetic correlations are similar suggests that we're going to learn similar things by studying pleiotropic relationships for common as opposed to rare variants. That might not be the same. That might not be true for every trait. So, like an example of this is, in autism, rare variants associated with autism will often cause intellectual disability, whereas common variants associated with autism are associated with increased cognitive ability on average. And so there could certainly be examples. I mean, there definitely are examples where we're probably going to learn different things and highlight different pathways by looking at common versus rare variant pleiotropic relationships. But I'm actually quite enthusiastic about applying, in particular, that last type of approach that I was talking about, the clustering type approach to rare variants, because the genetic architecture of rare variants looks a lot more sparse. So it seems like the heritability is much more concentrated in a relatively small number of genes. And the reason that is valuable for studying pleiotropy is it's just much more surprising when you observe a pleiotropic gene. Like common variants, can be pleiotropic, for example, because of LD, and there's just tons and tons of causal effects, such that, it's hard to interpret any one of them individually sometimes, but rare variant architecture looks quite different. So I think there's a lot of, there's actually a lot that we can learn by applying that type of approach to rare coding variants.
Yeah. Thank you very much. Yeah.
Thanks, Albert Henry from University College London. So I think in your talk, you mentioned that some examples that people do it kind of like hypothesis-driven analysis with one trait to another, but there are also like lots of hypothesis-free analysis where you just throw in a kitchen of traits and then just try to find which one are genetically correlated and causal to each other. Could you comment on, in such an approach, when people do it kind of in discovery mode, how would you then adjust for different power in these different GWAS summary stats, and how would you adjust for multiple testing?
Sorry, I missed the very first part. Are you talking about Mendelian randomization or just pleiotropy more generally?
I think about approach in genetic correlation analysis and Mendelian randomization. For example, when you do kind of like matrix for genetic correlations, how would you decide which one is genetically correlated and which one are not?
So if you just want to identify traits that are genetically correlated in some sort of high throughput manner, the standard way to do that is you estimate all the genetic correlations, you get p-values for them, and you apply some FDR threshold. And that approach works really well because tons of traits have overwhelming evidence for their genetic correlation, and that gives you, so you have quite a bit of power even after correcting for multiple hypotheses using the FDR approach. When it comes to causal relationships and Mendelian randomization, I just don't recommend applying Mendelian randomization in a high throughput manner. I just don't think it works.
How would you then, sorry, just follow up, how would you take into account the difference in different GWAS summary stats? They usually have different powers because they have different sample sizes.
Yeah, so there probably isn't any way to get rid of the sort of ascertainment bias that will result from different power among your traits. So you're not going to be able to do something like say oh, this trait has more genetic correlations than that trait when those two traits have really different sample sizes. But of course, these estimators of the genetic correlation are unbiased estimators that should give you, just basically what should happen is the standard error should increase. But it's not like the point estimates will like all go to 0 or anything like that. So that does enable you to compare genetic correlations across trait pairs with different amounts of power. It's just that, when it comes to something like identifying which ones are significant, then there's kind of no, way around the fact that you have different power among different trait pairs.
Thank you.
Hi, very great talk. I have a question regarding your remark that many of the causal variants might be unobserved, and I would be interested in what your suggestions are on how to solve this. What are options to address this issue?
So yeah, so many of the causal factors, like the phenotypes that are explaining the genetic correlations, are unobserved. So I don't think that we're ever really going to be able to observe those phenotypes most of the time. But what you can do, so maybe a concrete example would help. So, maybe there's just some signaling pathway that gets upregulated or downregulated at some, critical moment in development by all sorts of different genes. And, that the sort of specific activity level of that pathway, is responsible for all sorts of sort of developmental outcomes, including, I don't know, heightened CAD or something. We're probably never going to collect a massive number of individuals, in order to perform a GWAS, and measure that exact pathway's activity level. But we could still identify what it is, do you know what I mean? So we could still identify the genes that are involved, and do some experiments, actually do some biology to understand what's going on. So that's just an example of how you can, learn about causality with genetics without identifying, causal phenotypes.
Thank you.
Maybe I ask one about, have you tried to stratify by minority frequency the variants, potentially the causal ones, and run L-disco regression stratified by this? And did you observe a similar trend that rarer ones produce stronger genetic correlations?
That's a good question. So I haven't done that analysis, but I know someone who did in Hilary Finucane's lab. And I think this never ended up getting published because they had some powers. Basically, power was a big problem looking at low frequency variants. But I think that the main punchline of that analysis was that the genetic correlations were concordant, even though there were some interesting examples where it looked like there might be differences. And I think that basically what happened is that I don't think there was. I think that they just weren't sufficiently convinced that those differences were real to end up publishing it. But I think that was, if I had to guess, that is probably the case. It's mostly concordant with some that are different. And what we did do, though, is we did compare coding versus non-coding genetic correlations to see whether the difference we observed. Because, of course, we were comparing common coding correlations common variants which are both coding and non-coding with rare variants that were coding. And we didn't see a coding versus non-coding difference. So we think it's a rare versus common difference.
Are there any questions? Last chance? If not, then thanks a lot for this excellent talk.
Thank you.
And I hand over to Andrea.
Good morning, everyone. My name is Andrea Ganna, and I work at the University of Helsinki, and I'm co-chairing this session. Apologies for arriving a little late. So it's my pleasure to introduce Inga Prokopenko. She's a professor of E1 Health at the University of Surrey, and she will speak about the Atlas of Global and Local Pleiotropy.